Let's talk a little bit about Chapter 8 in our textbook. This is going to deal with early deterministic radiation effects. And we're also going to start to separate out terminology, talking about different kinds of early effects, different kinds of late effects, what this term deterministic means, why it's significant. Our textbook talks a lot about somatic effects, but we're also going to talk about the early effects on germ cells, right? Because we've made a distinction between somatic cells and germ cells. And so, I don't know, I find some of the wording in this chapter a little confusing, so I'll try to do my best to unpack some of the material and make it make a little bit more sense, hopefully. So here's some objectives. We'll describe the various early effects. We'll talk about acute radiation syndrome. We'll discuss the significance. So we've already defined what an LD 50/30 is. Now we'll talk about why that's important, as well as an LD 50/60 and why we have these two different kinds of lethality doses, and how that relates to organ dose limits. And then, like we did last week, we'll continue to kind of chart some of the effects to different organ systems. And for the early effects, the ones that I feel like are most important to us are the skin, blood, chromosomes, and germ cells. Okay? Now, I didn't just pick those systems out of a hat. I selected those body systems because they are the ones that, in terms of our work with this stuff, those are the ones that are the most common. Those are the things that we might see. Like, we will see people who have radiation burns from a breast cancer treatment. We will see people whose leukocyte count has been depressed because of a CT scan, right, or radiation therapy treatment, or a nuclear medicine study. With anything that we do as X-ray techs, or as nuke med techs, or as radiation therapists, we are going to cause chromosomal mutation, right? Any amount of ionizing radiation, any amount, can cause some kind of chromosomal mutation, and so that therefore has an impact on germ cells as well. So these are the ones that these are the systems that I feel like are most impactful to us. Chances are we're never going to see someone in a full-blown CNS syndrome from radiation sickness or a GI syndrome, but we may see people who we've depressed like a sperm count in because of radiation therapy treatments, or we may see people that we've depressed white blood cells because of CT or PET-CT imaging. So these early effects, these are the biological effects of radiation that occur soon after a human receives a high dose of ionizing radiation. So we're talking about high doses. And when I say high doses, I'm talking about stuff that we might only see in the therapeutic range, like 2 gray or more to the whole body. Not just to a selected area, like a targeted area, like what we do in radiation therapy, but a whole body dose of 2 gray or more. That's what I mean by a high dose of radiation. So a lot of the evidence that we have for this comes from fairly ethical research related to human populations after Hiroshima and Nagasaki, human populations after Chernobyl power plant. So our chapter talks about that, as well as animal studies that can be related back to human populations. Okay? Is there some unethical data related to this? Yes, there is. In fact, in our textbook, it doesn't point this out, but the data that we have related to sterilization, right? That data comes from the Third Reich, from the Nazis. Okay? So what they would do is they would have people who were Jewish or communists or homosexuals come in and register themselves as Jewish or communists or homosexuals. They'd say, fill out this paperwork, sit on this bench over here. And when they stood up from the bench, the next day they would have burns on their butt, basically in the area of their genitals. That underneath that bench, underneath that wooden bench, there was a radioactive source. Right? So what the Nazis had calculated was the amount of time that they would need to sit there and fill out the paperwork in order to sterilize them. Right? So that's a very unethical use of radiation, but I want to point out, because our textbook kind of elides that. How did we get this number? Right? The 6 gray or the 5 gray to cause permanent sterilization. It came from unethical research. Okay? Unethical research. So there is as we start to talk about early effects, we do have to differentiate between was this information ethically acquired or unethically acquired, okay? And we'll come back to that. The reason I'm stressing that now is I think towards the end of this class, we're going to have a class that's not in our textbook, but just talking about the ethics of this stuff. With all of your learning, what are appropriate and inappropriate applications of this stuff in your everyday life. But one thing that we can take some comfort in, I know that's kind of grim and grisly stuff, and I understand that. It's important for us to have a historical context for this stuff. Otherwise, we won't know why it's important now. So there's no questions on your test about Nazis or anything like that. But the thing I want to stress, and kind of the good news here, is on the whole, we are not going to see early effects of radiation in diagnostic imaging. Now, there are some caveats to that rule, and they are the ones that I've indicated, right? So we can see early effects related to blood cell counts dropping. We can see early effects related to burning the skin or epilation, right? But on the whole, we're not going to see these things like radiation sickness and stuff like that. But here's some of the somatic and genetic damage that we might see related to these early effects, and this comes from our textbook, page 159. So the factors that vary what kind of response the body will make to the exposure will depend on the amount of radiation the individual is exposed to, the ability of the ionizing radiation to cause ionization in the human tissue. And so what that's trying to talk about is like, is it high LET or low LET exposure? Like, what's its relative biologic effectiveness? Because like we've already learned, an exposure to an amount of alpha particles internally is going to do more damage than like an exposure to X-rays, right, of the same amount. The amount of the body area that was exposed. So are we talking about a whole body exposure, or are we talking about like a targeted area like what we do in radiation therapy? And then the specific body parts exposed, like we've already talked about quite a bit in this class. Each body system has different kinds of responses, and the various cells in that body system will have different responses too, depending on things like how, what their mitotic activity is and how differentiated those cell populations are. When we talk about somatic effects, again, somatic is anything related to the cells of the body, like the non-reproductive cells of the body. Cells that reproduce by mitosis, right? When we talk about the somatic effects, we're going to define that as the biologic damage sustained by living organisms after any exposure to ionizing radiation. So it's my personal damage that I received, right? Not chromosomal damage to germ cells that could be passed on to a subsequent generation, but this body system. So we're going to classify them as early somatic effects or late somatic effects, right? And this chapter largely deals with the early effects. So let's add another term in there, right? Deterministic, right? What does deterministic mean? Sometimes in the past this was called non-stochastic, so you should understand that these terms are interchangeable. Non-stochastic and deterministic are the exact same term, right? The way that I remember deterministic is that the amount of radiation dose determines the response, right? So the amount of radiation exposure determines a deterministic response, okay? So as the radiation dose increases, the severity of these effects will also increase. So a good example of this is like a sunburn, right? That is a deterministic effect of exposure to UV rays from the sun, right? And as I lay out there in the sun getting my suntan or whatever, I'm going to hit a threshold beyond which now I'm going to start to burn. And now the longer that I lay out there in the sun, the more severe that sunburn will be, right? So that's a deterministic effect. Another example of a deterministic effect, right, is y'all are at a point in your college career and within my classes. If I were to say, okay, We've all arrived at this moment where it's called quarter day, and Uncle Benny is going to give every single person in here a quarter, right? The fact that you've made it to this threshold in my class means you receive a quarter, right? That would be another deterministic effect. You've been in my class long enough, now you all get a quarter, right? Now let's compare and contrast that with, now if I said everyone flip your quarter, right? Everyone flip your quarter, and everyone who has heads, you get to stand up, right? That is now not a deterministic effect. We're gonna call that a oh my goodness, I just right out my mind. That's gonna be like a randomized effect or stochastic effect. Okay, randomized or stochastic effect. Okay, so there are things that are stochastic effects in radiation. The biggest one being cancer, right? That's the coin toss. Every time I'm out there in the primary beam in X-ray, or every time I'm around the neutron contamination in therapy, or every time I'm around the radioisotopes in nuclear medicine, I'm flipping that quarter now. And if I get so many heads, eventually the likelihood of me getting cancer is pretty high, right? So that's just a compare and contrast. But what we're talking about is just the deterministic effect. So as the dose increases, the severity increases, and these results have a threshold. So at some point we pass that threshold and we're going to start to see the incidence increase. So here's what that threshold looks like. And this is something that you should just go ahead and commit this curve to memory, right? Some of us have probably already seen this curve, but we're going to call it a threshold nonlinear dose response, right? It is not a line, it's curve shaped, right? And it has a threshold. It has a point below which we do not see any of these responses. So again, a good example would be, let's just keep with the skin burns, okay? Let's keep on giving people skin burns today. There's a point at which beyond that we start to see a skin burn. But any dose prior up to that point, we won't see the skin burn, right? Now, that threshold will be different for every single person that's exposed to the radiation. And it has nothing to do with whether or not they get a sunburn, right? Folks in radiation therapy will tell you this. Like, I've seen folks as pale as me do just fine in terms of radiation exposure. And then I've seen African folks who can be out in the sun a lot who burn quite quickly when exposed to radiation. So I just want to point out our metaphor is helpful, but it has its limits. We are talking about a different process in terms of what radiation does versus UV rays. But there's a threshold that they've identified for, a large percentage of the population. Beyond that threshold, we will start to see the incidence and the severity increase. Okay? So that is a threshold nonlinear dose response. And all of these early deterministic effects of radiation are pretty much modeled by this graphic. Okay? All of these early deterministic effects are threshold nonlinear dose responses. So, why do we call them early deterministic effects? They will appear within minutes, hours, days, or weeks of the time after radiation exposure. Okay? They require a substantial dose of radiation. So I've already said by substantial I mean 2 gray or more, or higher. Right? We'll put an arrow for higher. So who here exposes patients to 2 gray or higher? Does anyone in this room expose patients to 2 gray or higher? Therapy, y'all's hands should be up. I think some nuke med, like I think PET CT, has this potential. But for the most part, therapy is the big one. So some things that could produce early deterministic effects would be radiation therapy, interventional radiography. Those are the two that stand out to me. And I know Mr. Al-Shafi loves interventional very much. But one of the reasons, I want to just point this out, he last week he was talking about why the heck am I even in this place, right? Well, one of the reasons that we are in interventional, right, largely is to be whistleblowers, because we've received this training where honestly the cardiologists and some of the other people working around this, like the urologists, have not received this training. they're not up on the fact that the machine that they're using could be causing skin burns or chromosome damage. They're not quite familiar with the severity of some of the things that they're choosing to do. And so we can provide that kind of reinforcement to say, hey, we just hit the 10-minute fluoro timer. Let's try to wrap things up. That's why we get paid the big bucks in interventional for just sitting around and playing Minesweep. Okay. Early deterministic effects. When we pass that threshold of 2 gray and we get up in the area of 6 gray, we're talking about things like nausea and fatigue, erythema, epilation, blood and intestinal disorders, fever, dry and or moist discrimination, and temporary or permanent sterility, right? So I put parentheses genetic effect there, right? Because what I'm talking about is damage to the testes, which is a somatic organ system, but within the testes or within the ovaries, there are germ cells that are responsible for reproduction, right? So here is an example from our textbook of a fellow in an awesome sweater, which I would love to have his sweater. But he sustained a significant erythema to his forearm. That is not from exposure to fire or anything. That is simply radiation that he was exposed to as a result of the nuclear power plant accident at Chernobyl. Now I want to point out one thing about Chernobyl, because I know probably at this point in this class people are starting to ask some questions about what is nuclear power, how does it work, and we will have an opportunity to watch some of a documentary about it. But Chernobyl was unique in that it was not just a nuclear power plant, they were also using the refinement processes to produce nuclear weapons, right? So it was unique in that also it had no containment. It had no way to contain the nuclear radiation in the event of an accident. So when we look at images of Chernobyl and when we talk about Chernobyl, what we're seeing is an uncontained nuclear contamination, right? So the firefighters and people who responded to Chernobyl had such severe responses to the radiation there because they were dealing with an uncontained nuclear power source, right? So I've read, there's a book called Radiation that's written by the doctor that actually responded to Chernobyl. He's a British fellow. I think his name's Peter something or other. I can't remember his last name. But he is the one who kind of pioneered the bone marrow transplants that wound up saving some of these people's lives, right? And we'll talk about why that's important in just a second. Let's talk about acute radiation syndrome. So what exactly happened to these people at the power plant as they were exposed to the radiation from the accident? There's a number of stages of response, and all patients, regardless of the syndrome, experience the same stages. Okay? So the length of the stage will vary based on the amount of radiation exposure. But there's prodromal, latent, and manifest illness. Here are some dose thresholds, and this comes from our textbook also. So at about 1 gray, we'll start to see, and this is page 164 in our textbook, about 1 gray, we'll see initiation of a prodromal phase, which is really fun stuff like nausea, vomiting, diarrhea, fatigue, leukopenia, those kinds of things. Your body, like your midbrain, is saying, I don't know what the heck just happened, but I think I was exposed to something that made me feel sick. It initially thinks that you were exposed to like a pathogen, right? Like a bacteria or a virus or something like that. And so it responds to the radiation exposure like it would to that virus or to that bacteria. It makes you puke, right? Which is the way body gets rid of bacteria and viruses and stuff, right? So that nausea and vomiting is the body's natural response to exposure to something that makes it ill. Then it goes into a latent period where any amount of time, it could be a matter of weeks to a matter of minutes, depending on the radiation exposure amount, this acute radiation exposure, where nothing happens. right? It's kind of the calm before the storm. So we'll call that the latent period. Then depending on what kind of exposure the person received, they will move into one of these syndromes or possibly a combination of these syndromes, right? And so we'll call that the manifest illness. And then the body will either shut down and die or it will recover, depending on a number of factors. So here's another way to think about it. So we have prodromal, latent, manifest illness, and death. And I think I told you the mnemonic that I use to remember these and the order in which they come is please let me die or recover, right? Please let me die or recover. And this is just another slide. I've just added this in here just because there's a number of different ways we can think about this. But we see an increase in symptoms over time, and then a latent period where no symptoms increase, and then a manifest illness, and then this point at the curve on the far right is either the death or recovery. Now, if we have a significant enough exposure, the recovery can be slightly hampered, right? There can be atrophy of the organ systems and permanent damage that can occur. But for the most part, people can fully bounce back from like a hematopoietic syndrome. So let's talk about that one. This is doses between 1 and 10 gray will trigger the hematopoietic syndrome. And this prodromal stage will initiate a few hours after the exposure. And it will persist as long as 3 weeks. In most populations, it's a matter of days, like 5 days, 6 days, that the prodromal phase remains in that person. It does depend a little bit on how healthy the individual was at the time of exposure. And the problem with this is if we're talking about a whole body exposure that has depressed white blood cell and red blood cell counts, is the risk of infection. So the term for that is pancytopenia, which means like whole body destruction of blood cells. Pancytopenia is just the fancy name for whole body destruction of blood cells that can result in hemorrhage, especially internal bleeding within the intestinal tract, and infection because of decreased white blood cell counts. So if we're talking about the risk for death here, 2 gray, if we have a very sensitive individual, they may die after about a month, 6 to 8 weeks. Okay? 4 to 6 gray is the range of the LD50-30 for this exposure. 4 to 6 gray is the LD50-30. After 10 gray, we're talking about death within 2 weeks, and we'll also have the addition of both the hematopoietic syndrome as well as the next syndrome. And in that, we're talking about the GI syndrome. So GI is 10 to about 100 gray exposure, and death is independent of the dose. Anyone who has received an exposure sufficient to cause the GI syndrome will not live. We can bring people back out of the hematopoietic syndrome through those bone marrow transplants that were pioneered at Chernobyl, right? And the reason I want to kind of talk about that a little bit is it changed some of the way that we think about this as medical responders to this stuff. Chernobyl changed it because up to Chernobyl we looked at the LD50-30 and we said this is what determines the amount of lethality of the dose is the LD50-30. How did 50% of the population die after this exposure? That's going to be our baseline. After Chernobyl, with the advent of these bone marrow transplants, some of them were successful. And so they looked then to the LD50-60. How many people survived after 2 months thinking about this medical intervention? Okay? So you'll see in our textbook sometimes it says the LD50-30 or with medical intervention the dose might be slightly higher. Right? Once we get to the GI syndrome, we can go back to talking about LD50-30 because anyone who receives this amount of dose will be dead within 30 days. Right? So folks die more or less around the same time. We're talking 3 to 10 days without medical intervention, 2 weeks with medical intervention. I think if I was exposed to this radiation amount, I would just say intervene with a gun because I am done, right? This sounds really like a struggle because the death is a result of intestinal denuding. Why am I talking about this morbid, terrifying, horrible stuff? Okay? Well, because it is scary stuff. Hopefully it'll help us remember some of how these organ systems work and what we were talking about last week with the different types of responses of cells to radiation. Like I'm talking about the law of Burgundé and Tribandot, right? What happens inside of the intestines, right? If you look in our textbook, let me find the illustration. Yeah, sure.
The book says that the dose for GI is 6 to 10 gray. So is it that or is it 10 to 100? Because that's a huge difference.
It is. What this is, okay, we can add 6 in here. So 6 to 10, we have both hematopoietic and GI, right? From 10 to 100, we just have GI. CNS kicks in at, I think, what is it, 50 or higher? Yeah. The significant thing here is these syndromes blur into each other, right? So rather than memorize what these numbers are saying, because you will look at different charts and they will have different numbers on them, and that's fine because they have different findings. The significant thing here is if you look at page 161 in our textbook, let's park it here for just a little bit because this is the takeaway here. It's not necessarily what amount causes what syndrome, right? If I ask you a question about a person is exposed to X amount of radiation, what syndrome will they be in? They will be a very obvious amount. Like a person is exposed to like 2 gray, what syndrome could they wind up in? Right? Right, good. Versus like if a person is exposed to like 10 gray or more, what syndrome could they wind up in? Yeah, that's all. But the big takeaway here is asking why these different syndromes occur and why the heck is there a latent period? Right? So let's talk about that real quick because this ties right back to what we were talking about last week with the law of Burgundy and Tribandot. So page 161 in our textbook, you see healthy epithelial lining of the small intestine. GI syndrome is most closely related to destruction of the small bowel, right? So what's happening? It talks about after the epithelial lining is exposed to radiation, we have denuding of the cells, okay? And this is the exact same thing that happens with that skin burn. The reason the skin is red is because the mature cells have died, right? We've lost some of the mature cells, and we've also damaged the stem cells below them, the less differentiated cells that were going to eventually become mature. That's why there's this latent period too. So let me try to draw this out. So it's drawn in our textbook that there's these folds, and let's imagine for the purposes of what I'm talking about, we would divide one of these folds into levels of cell maturity. Okay? So this cell up here is very mature, right? And this cell down here is very immature. Because this is kind of how a lot of body systems work. You've got some base level of immature cells, and as they trickle up to the level of the skin, or as they trickle up to that internal part of the small bowel, they are now very mature. They've matured in the process of getting up to that area. And now they're doing whatever function they were supposed to be doing there in the small bowel or there on the skin, right? But what does radiation biology tell us about the difference between these levels of maturity? Which one of these is most radiosensitive? The immature one, right? The immature thing is the most radiosensitive. So when it's exposed to radiation, right, all these immature ones will die because they're more radiosensitive. They're going to die. Okay? Now the mature ones are still there. They're kind of reeling. They're trying to figure out what's going on. They might go into a slight delay to kind of repair, but they are more radio resistant. They can repair. So they repair and they continue to do what they do until what happens? They reach the end of their cell life cycle. Right? And now when they die, there's nothing there to replace them. When they die, there's nothing there to replace them. So this is why there is a latent period. This is why I'm stressing these syndromes, is because they let us see real hands-on how law of Burgundy and Trivendo affects these organ systems. Because in each organ system of your body, there is this process. Except for one, I guess like with the CNS, we don't see this process, right? The brain cells are just the brain cells, are just the brain cells. They're not going through this replenishment process. But the skin, the small bowel, especially the germ cells, the sex cells, have this move from immature cells up to increasingly mature cells, right? So again, the concern with the hematopoietic syndrome isn't necessarily the circulating mature blood cells, right? It's the bone marrow. It's the cell death that occurs with the immature cells in the bone marrow, and then once the mature blood cells die off, there's nothing there to replace them. So without a bone marrow transplant, this person will probably die after a period of latency. And that is why with the CNS syndrome, at doses over 50 gray, we no longer see a latent period, or the latent period will be very, very short, right? Because now we're talking about the direct death of a cell system that has no replenishment process, right? So death will occur in 2 to 3 days. They don't even enter into the hematopoietic or the GI syndrome because there's been enough destruction of just very mature nerve cells to where the body just shuts down. The brain is no longer telling the heart to beat or the lungs to breathe. And if you look right here, autopsies reveal little cellular damage. So this is just kind of one of the mysteries of radiation syndromes, and the only reason I'm stressing it again is so that we understand the dynamics of these cell replenishment processes and how that relates to like law of Burgundy and Trivendo. So let's talk a little bit about this LD. I know we've talked some about LD 5030 already, and we've said it's the whole body dose that will cause 50% of the population to die within 30 days. We use this as a measurement, and it's been fairly accurate, especially when we do animal experiments and stuff like that. And the dose for us is about 3 to 4 gray. About 3 to 4 gray, and this is a whole body exposure. 3 to 4 gray is our LD 5030, for the general population. Here's another way of representing this, and we said, what kind of curve is this? Threshold, right. And if you look, the threshold actually initiates around 1 gray. So there's infirm people or people who are incredibly radiosensitive. After 1 gray of exposure, we start to see an increase of people dying. But the actual LD 5030, if we trace this 4 up and this 50 over, we hit right there. That's the actual LD 5030, where 50% of the population is dead after an exposure to 4 gray. This shoulder region up here, we said, is the point of overkill, right? Where the LD 5030 is now 100%. We can't kill more than 100% of the population. But it also is helpful to think about an LD 1030 or LD 5060, LD 160, right? These are other measures of lethality. And I should point out these LD 5030 and stuff like that, it's not just radiation that scientists talk about this with, right? There's LD 5030s for inhaling gas fumes, right? There's LD 5030s probably for Lysol. Right? There's LD 5030s for just about everything that we make that could potentially cause damage to the body. Right? So chemicals, all sorts of things that have LD 5030s. The LD 5060 may be more accurate. And the reason I say that is, again, post-Chernobyl, with the advent of these bone marrow transplants, the LD 5060 gives us a better picture of how effective the medical intervention was, right? Because if they're alive 2 months after that exposure, chances are they'll keep on trucking. Their body has moved into a period of time where they can start to repair the stem cells that were lost due to the radiation exposure, and they will move forward with their lives, right? There may be some atrophy of some of the organ systems and things like that, but chances are they will heal. Like the smiling fireman with the awesome sweater, right? So this repair and recovery process is exploiting the repair enzymes that we talked about within the cell, right? And immediately after irradiation, the cells begin to repopulate and repair themselves, especially the mature cells. And the amount of function damage determines the organ's potential for recovery. If there's severe damage to the organ, it may experience some atrophy or scarring related to the exposure. One of the things that's interesting about this process is we talked about how oxygen can enhance the radiation damage. Oxygen also serves to enhance the repair process. So there's kind of a double bind there, where we've said that hypoxic cells are more radio resistant, but they also will not be able to repair as quickly, right? So in terms of repairing, the chances of repair, well-oxygenated cells are more likely to recover, okay? To this point, and this is one of the reasons why we say this, repeated radiation and injuries can have a cumulative effect, right? Because of what we're talking about, this repair process. There's going to be some of the stuff that can't be completely repaired, or there could be atrophy, and so another large radiation exposure could kind of work towards a cumulative effect. There is approximately 10% of radiation exposure that's not repairable, okay, once we get into these levels of 2 gray or higher. But the remaining 90% may be repairable over time. So, local tissue damage, when we look at, for example, on page 160, the woman who sustained burns to her back, these can be a destructive response to high radiation doses. And it can lead to atrophy of the organs and tissue. They may not be able to function. Listen, radiation therapy is one of the reasons we try to avoid treating like the heart when we're doing a breast treatment, or we try to avoid treating the lung, right? Because we could potentially cause atrophy to those structures that would be irreparable. And those are significant life-sustaining structures. So let's talk a little bit about these skin effects. There's radiodermatitis, which is cancerous lesions, and this is one of the historical evidences. So this was some of the early radiation workers experienced this with repeated high-dose exposures. Okay, epilation, or loss of the hair, sometimes called radiation alopecia. So moderate doses of radiation may cause temporary hair loss, and then large doses can result in permanent hair loss. So in the radiation therapy field, again, a lot of times we might go ahead and counsel our patients if we're doing a whole braid treatment, go ahead and shave your hair, right? Because you're going to lose it all, and it's probably not going to come back. Occasionally it'll come back, and I had one patient, their hair came back and it was curly afterwards. So I don't know why that was the case, but it can come back. It's going to depend again on how that person's body responds to that radiation amount and what exactly that amount was. Then desquamination, again, we see this in radiation therapy. We see this perhaps with high fluoro doses, where we actually burn the skin such that the skin sloughs off. Right? So we talk about moist desquamination and dry desquamination, right? Moist is that initial response, and then dry is what follows. Hematologic effects. So if we have a whole body ionizing radiation dose as low as 0.25 gray in tissue, we can depress hematology. We can depress the blood cell count and the white blood cell count. This is significant because that's a dose in the range of CT. Right? Now, most CT scans, I'm not giving a person a whole body exposure. Right? But occasionally I do need to do a CT scan like of the head, neck, chest, abdomen, pelvis. Or in nuclear medicine, we might do a CT scan, or a PET CT scan that includes all of the head, neck, chest, abdomen, pelvis. That person has received a dose that is large enough to cause hematologic depression. So their white blood cell counts will be low, and their red blood cell counts will be low for about a month after that exposure. And as we pointed out, that response is not going to happen immediately after that exposure. Right? There will be some damage done to the mature cells that are circulating in the bloodstream, and we've pointed out that the lymphocytes, even though they are mature cells, are among the most radiosensitive cells in the body, right? Even though they are mature cells. But what's significant is this stem cell right here, right? The one that could go in any direction within the bone marrow, right? When the stem cell is damaged, it is much more radiosensitive. Right? So it will be a period of weeks or maybe even as long as a month before we start to see the effects of that initial stem cell damage. So just like what we talked about earlier with the GI syndrome, this hematological effect will have a latency period. Right? It will have a period in which there's no observable effects. And then after that period of time elapses, now we'll start to see, okay, here's the manifest pattern. Right? As those stem cells have evolved into more differentiated, highly specialized cells, now their absence is marked. This is especially significant for radiation therapy because we will need to monitor people's white blood cell counts and red blood cell counts as they go through the course of their treatment. We will not see those numbers fall off immediately. There will be a series of weeks, perhaps, when all of a sudden the numbers could just plummet, right? And we'll almost need to be tracking those patients every single day. They'll need to go to the lab and get a blood cell count before we can do their treatment, right? Because we are towing that line with radiation therapy and with some chemotherapy treatments between killing all the cancer and also killing all the good cells too, right? So this is significant to folks in radiation therapy. As I mentioned already, this is significant to us in diagnostic imaging insofar as with CT, we have the potential to trigger this. Cytogenetic effects. So now we're talking about the chromosomes of these cells, right? So up until this point in the lecture, we've been talking just about damage to the entire cell that causes cell death. Now we're zooming in even closer on the cell, and we're looking inside the cell membrane, or inside the cell nucleus now, at what happens inside that cell nucleus after radiation exposure, right? What happens to those chromosomes? So cytogenetics is the study of cell genetics with an emphasis on the chromosomes. And we create these maps called karyotypes, and typically we do that during metaphase. So metaphase is very, very important to us anytime we're doing some kind of radiation biology study, because when we freeze cells in metaphase, we can actually look at the chromosomes and determine if there's any kind of clumping going on, or like a ring shape, or fragments of chromosome, all of those things that we kind of charted out earlier in last lecture. This is when they become apparent, is when we look at the cells under a microscope during M phase. So here actually is a karyotype from our textbook. So again, these were chromosomes that were captured and mapped during that M phase. And we can see different kinds of damage that has occurred at this time. So let me try to point out some. So, and also we see the difference between, for example, a male patient here and a female patient where there's the 2Y, or the 2X, I'm sorry. But yeah, this is I don't know if y'all remember earlier in the trimester when I had images of like astronaut blood after they've been exposed to radiation. We could see different kind of colored fragments. They can additionally color these karyotypes to better enhance different levels of chromosome damage. All right, this is one of our last points, right? So we've mostly been talking about somatic effects, these early somatic effects that which we said are all deterministic in nature. So the amount of radiation is directly tied to the severity of the effect. Now I want us to switch one switch in our brain, and we're going to talk about early, like germ cell effects. These can also be deterministic. Okay, these can also be deterministic effects. And how do they function? Well, exactly like the early effects function for somatic cells, in that we have stem cell type things in these primordial follicles and spermatogonia, right? And anytime we have testes or the ovaries are exposed to radiation levels over 2 gray or higher, we can start to see this effect, right? And so we are talking about the potential for either temporary or permanent sterilization of this individual because of this effect. Now, where do we see this in medicine? Again, it's radiation therapy. Anytime we are treating folks for testicular cancer, we are thinking about this potential, right? So, interestingly enough, the initial, again, there will still be that latency period because the sperm cells in the ovum are highly differentiated, very mature cells. So they are radioresistant, they are prepared to repair themselves. They can carry on after an exposure, right? So 2 weeks out, then we start to see the sperm cell count decrease. Or we see delayed menstruation for females, okay? And it all stems back to, this is an early germ cell response to a damage that occurred with a more radiosensitive population that was less mature, okay? Now, why is this significant and why am I stressing this? Well, let's differentiate just a little bit because I said that there can also be these chromosomal damages that can occur, right? And this is actually where we should all pay attention and think, okay, this is significant, right? Because as far as we know, the chromosome damage stuff is completely stochastic. It's completely randomized. There's no threshold dose, right? There's no threshold dose for the chromosome damage, right? So what does that mean? Well, if I expose someone to radiation, like for example for a CT scan or for a PET CT scan or even for an X-ray, right? If the testes are exposed, I may not see this early genetic effect where the sperm cell count drops, but I may see chromosome aberrations, right? I may see chromosome aberrations. There is a potential to cause chromosomal damage inside of these cells that the cell could then repair itself, but not repair that chromosome damage and pass on that damage to a subsequent generation. This is the reason, this is one of the big reasons why we practice occupational radiation safety, right, in terms of the people who are being exposed. And if you have a younger person, male or female, who you're doing a CT scan on, it is not inappropriate to tell them, look, if you're thinking about having kids, I would wait a month or two, right? Why would I say a month or two? Well, again, because of this. I'm waiting for that cell population to repopulate. And if it repopulates in a way that's sufficient to repair all the damage that was done by the CT scan, then the likelihood of passing on that chromosomal damage to a subsequent generation is significantly decreased. Okay? I have told people that. I know it sounds kind of crazy, but if it just depends on the population you're working with. But if you're working with younger professionals, and you can ask them, hey, are you starting out? Are you just having a family? You can get a feel for where people are at in their life, right? And you can let them know, you've received a radiation dose today that could potentially cause chromosomal damage. I would encourage you to wait on having kids for a month or two in order to just, it's a very, very small risk, but just to further minimize that very, very small risk of passing on that chromosome damage. Does that make sense? Okay. That